In this video, we will discuss bacteria and its morphology. To begin with, we can talk about the larger group to which bacteria belong, prokarya. Cells can be broadly categorized into two groups, prokaryotes and eukaryotes. Prokaryotes are further divided into two domains, bacteria and archaea, which are single-celled organisms. I include an asterisk here because though prokaryotes are considered single-celled organisms, there has been growing evidence that some groups of prokaryotic cells can organize and form structures that resemble and function like multicellular organisms. Also note that while both lack a nucleus, bacteria and archaea have separate evolutionary lineages with a number of physiological, metabolic, and genetic differences. Usually people are most familiar with the disease-causing pathogenic types, for example, Streptococcus bacteria, which causes strep throat, as well as cholera or tuberculosis. These make up only a fraction of the large diversity of bacterial species on Earth. Bacteria grows in many different habitats and conditions, including deep oceans, as well as frozen ice. In this center photo, you can see ice bubble assemblages containing living bacteria near the surface of a lake in Antarctica. They also live in the guts of many different animals. In the example of vultures, their unique gut microbiome includes bacteria that help them safely consume and remove rotting carcasses. In addition to its role in human physiology, one of the most important roles of bacteria includes helping in the decomposition of organic carbon. That includes dead and rotting organisms, and this is vital to the ecosystem health. Now we are going to discuss how to distinguish and classify different bacterial forms. Normally, bacteria are transparent unless they are stained. Please refer to the bacterial gram staining video for more information on this process. Some species of bacteria exist in loosely arrayed filaments or colonies. In the figure you see on the bottom right, you can see bacteria on a human epithelial cell. It is helpful to understand the relative size of bacterial cells by comparing them with eukaryotic cells and the nucleus in this image. Bacterial cell structure is generally simpler than that of many other organisms, as there is no nucleus or membrane-bound organelles. Due to the presence of a rigid cell wall, bacteria maintain a definite shape, though they vary as shape, size, and structure. When observing bacterial cells, we can identify three basic cell shapes. The first is spherical, also called Coccus. The second is rod-shaped, also called Bacillus. And the third is spiral, also called Spirillum. In the first form type, Coccus, or cocci for plural, these are round cells that sometimes appear slightly flattened when they are adjacent to one another. Most cocci are approximately 0.5 to 1 micrometer in diameter. They have several distinct arrangements based on the planes of their division. When a cell has a division in one plane, this produces either a Diplococcus or Streptococcus arrangement. In the top two images, you can see on the left a Diplococcus arrangement in which cocci are arranged in pairs, and on the top right you see a Streptococcus arrangement in which the cocci are arranged in chains. These two images are examples of how the bacterial cells may look under a microscope. On the bottom of this slide, we see a cell that's divided into two planes that produces a tetrad arrangement. This is called a tetrad arrangement in which cocci are arranged in squares of 4. Again, these two images both show a bacterial cell in a tetrad arrangement under a microscope. When a bacterial cell of Coccus form is divided into 3 planes, this produces a Sarcina arrangement. A Sarcina is a cocci that is arranged in cubes of 8, as you can see in the top two photos in this slide, the top left and the top right. And the final arrangement for Coccus bacterial forms is when division occurs in random planes that produces a Staphylococcus arrangement. Staphylococcus is cosi arranged in irregular and often grape-like clusters. You can see two examples of this on the bottom of the slide. On the bottom left is what you would see under a microscope with gram-stained bacteria, and on the bottom right is a much more magnified image. The second form of bacterial cells is the bacillus, or rod. A bacillus is a rod-shaped bacterium that has three different types of arrangements. Average bacillus are usually 0.5 to 1 micrometer wide by 1 to 4 micrometers long. In the top right, you see an example of a bacillus form, a bacillus arrangement. This is a single bacillus cell. In the middle left, you can see the arrangement of streptobacillus, which is a bacilli, which are multiple bacilli, that are in chains. And the third arrangement for bacillus cells are the Coccobacillus, which you see in the bottom right example. And these are bacterial cells that are oval in shape and look similar to what you might see for bacteria that are in Coccus form. The third type of bacterial cell shape is Spirillum. This is also called Spirilla or Spirillum for a single cell. These are curved bacteria, which can range from a curved shape to a corkscrew-like spiral. These spirals usually range in size from 1 micrometer to over 100 micrometers in length. Within the Spirillum bacterial cell shape, there are three forms. On the top right, you see a possible form in Vibrio, which is a curved or comma-shaped rod. The top right image shows you what this may look like under a microscope. In the middle left, you see what the Spirillum form looks like. These are usually bacterial cells that are thick and rigid spirals. And the third form is spirochete, a thin, flexible spiral bacterial cell, which you can see in the bottom right picture, is a spirochete species called Borrelia burgdorferi. This is the bacterium that causes Lyme disease.